We have a bunch of members already on, so I think we should go ahead and get started. Thank you so much for joining today. A reminder, the public webinar series is designed to educate attendees on the scientific ecosystem, and each month we'll have a representative from a data coordinating center and new developments from the centers that support them. Today, I'll share some KC scientific updates, upcoming events, and we'll hear from our featured speakers. Just a reminder, if you're unable to attend last month's webinar or webinars prior, all the recordings are available on our YouTube page with this link here below. As the KC, we'd love to create use cases around how you, the researcher, use data sets within the DCCs. Here's our landing page for the KC. So if you have any feedback, questions, or would like to be added to our listserv, please email us at help@cfdeknowledge.org. Earlier this month, we were invited to participate in the Boston session of the Festival of Genomics. Here are some pictures from that session. It was wonderful to meet people in person, and the invited talk that we had in the Genome Dome gave us a chance to showcase CFDE as a whole, represent both the KC and DRC, and even include 4D nucleome DCC. So this just marks the first of many conferences that we'll be doing together, but we'd like to know if there are any others you think that we should be attending as either the KC and DRC, just to showcase CFDE overall. But with that in mind, later this year, we'll be attending an even larger conference in genetics and genomics, the American Society of Human Genetics. Our booth will also be featuring DCCs, including GTEx and hopefully Kids First, and any others that will be joining us. Please let us know. We'd love to collaborate. We'll also have the centers and the CFDE there as well. And we are also working on hosting an ancillary session that will take place during this event as well. There are some other events that will be within the ecosystem that we'd like to socialize, including the Metabolomics Workbench Virtual Summer Workshop in July, the IDG CFDE Specialized Course in July as well, and the Kids First CFDE Pediatric Power Up in August. These event details will be publicly available via these slides and also on our Twitter. Today we have a scientific presentation from Illuminating the Druggable Genome, or IDG, Data Coordinating Center. Before we hand it off to them, I'd just like to remind you that IDG is one of the 14 resources within the Common Fund data ecosystem. IDG is one of our more mature resources within the ecosystem, and they're also a consortium aimed at highlighting current knowledge of protein targets and further studying the function of specified understudied targets. One of the resources that they'll walk through today is Pharos, a comprehensive integrated knowledge base for the druggable genome to illuminate the uncharacterized and/or poorly annotated portion of the druggable genome, focused on 3 of the most commonly drug-targeted protein families. Drug Central is another resource that will be touched upon today, which is a drug information resource created and maintained by the Division of Translational Informatics at the University of New Mexico in collaboration with IDG. So one thing as the KC is we'd like to bring both the IDG consortium and DCC results to the user. Actually, IDG was really easy for us to incorporate in the KC, and we have their summary representations in our beta portal. So if you were to search for a gene of interest in our search box on our main page, as you can see here, users are brought to the corresponding gene page where they can view the Pharos drug target level classifications, as you can see here. These classifications are clearly marked exactly as they're represented on Pharos to summarize the amount of evidence supporting the gene as a potential drug target. We also provide a link to the Pharos UI, as you can see here, to further explore the data. This card on the gene page also puts the results in context of other gene information summarized by the other DCCs. And instead, if you're interested in searching for a disease like bipolar disease here, the disease page also includes Pharos target drug development classification as represented on their site, showcasing which genes are potential drug targets for the disease of interest. We are working to develop new analyses that can also be represented in future versions of the KC as well. I'd now like to hand it over to today's featured speakers from the Illuminating the Druggable Genome IDG Data Coordinating Center, and I'll pass it off to Jeremy.
Thank you, Mackenzie. Let me start sharing my screen here. Is that looking all right?
Yes, thank you.
Okay. Yeah, thanks for organizing this webinar series, and to the KC, of course, and thanks to your team for integrating our data as you have so rapidly, and that really was great to see at our annual meeting in March. Okay, we're really pleased to be presenting today, so hello to everyone who's in attendance or perhaps watching the recording. We are going to give a team presentation. There's 4 of us. And we're pleased to participate in this webinar. Thanks to the Knowledge Center and other CFDE leaders for inviting us to present this overview and highlights, as we're calling it. My name is Jeremy Yang. I'm a co-PI of the IDG Data Coordinating Center, DCC. My fellow co-PI is Christoph Lambert, and both of us are at the University of New Mexico in Albuquerque. Also presenting with me today are Anna Waller, scientist and project manager for the IDG Knowledge Management Center, KMC we call it, and the Resource Dissemination and Outreach Center, RDOC. So she did a lot for both. Jamie Holmes is also presenting in our team here. He has been project manager and developer of key resource Drug Central. And our 4th presenter is Tim Shields, project manager and developer of PHAROS of NIH NCATS. Anna and Jamie and I are at the University of New Mexico and have been IDG contributors since its beginnings over a decade ago. Tim is at NCATS and joined IDG a few years in, taking over from Rajarshi Gua, not an easy task. Raja moved from NCATS to a senior position at Vertex Pharmaceuticals, so he's done very well. This webinar has 3 parts. First, some history and background, which I'll do. Then Anna Waller will present on the importance and impact of IDG over its lifespan. In part 2, I'll present an overview of the many IDG data resources. Then Jamie will present Drug Central, an exemplar and featured IDG knowledge base. In part 3, Tim will present PHAROS, the IDG data portal. Given the time allowed today and how much is in IDG, this presentation will really just scratch the surface, but hopefully be a reasonable length so that you can so it doesn't drag on too long either. So hopefully we're being concise, but quite informative. But really, this is only going to begin to introduce the IDG program and its data resources, which we believe are of much relevance to CFDE and potential value for biomedical data science use cases, integrating common fund and other data sets, which is kind of our overall theme, of course. However, for those interested in learning more, we will provide suggestions, links, and references to an extensive corpus of documentation and publications. And please feel free to ask questions via the chat or chat via the chat amongst yourselves. But there's also the Q&A, as Mackenzie mentioned, for asking questions of us, the panelists and the presenters. And of course, you can ask questions after this webinar by contacting us, so please feel free. And this is the outline of the webinar, as I described. The IDG program was designed, as Mackenzie indicated, to improve our understanding of understudied proteins from the most commonly drug-targeted protein families. 1, G protein-coupled receptors, 2, protein kinases, and 3, ion channels. 2, catalyze research in understudied genes that have high potential to improve clinical therapeutics and human health. The NIH Common Fund sponsored IDG from 2014 to 2023, supporting a diverse array of experimental and computational research. Druggablegenome.net is the IDG website, and PHAROS is the data portal, integrating many other data resources from IDG across NIH and beyond. Bottom left on this slide from the druggablegenome.net is a graphic which illustrates the extensive IDG network of institutions and collaborators spanning the US and including European collaborators, EMBL, EBI in particular, and also University of Copenhagen. Bottom right is from an IDG Toolfest event with a sampling of logos from IDG discovery tools and data resources. Like any successful effort, the key ingredients of IDG have been the dedicated and capable persons doing the work. This slide includes some of the PIs and leaders who brought to IDG a strikingly diverse array of expertise, spanning foundational basic sciences, such as, for example, kinase research from cancer biologist Gary Johnson of UNC, GPCR research from neuroscientist Brian Roth, and the computational informatics and data science capabilities of the Knowledge Management Center, Research Dissemination and Outreach Center, KMC and RDoc, and the Cutting Edge Informatics Tools groups. Tudor Aprea was the PI or API of the KMC and RDoc, conceived and led many of the computational innovations, including the Target Development Level TDL classification system. And Tudor was at a UNM before retiring to emeritus status and moving on to a senior leadership position in industrial pharma, very much AI-based. Now, since this is an CFDE webinar, it is especially fitting that we recognize Avi Mayan, pictured here as well, who is PI of the CFDE Links, DCC, and DRC, and who was a PI for the IDG, and in whose group developed a great many of the IDG data resources featured in this presentation. The IDG program was focused on the human proteome, and more specifically, the three most common drug-targeted protein families, as mentioned, and their protein coding genes. However, two other key entity types were prioritized as central or essential to this mission. Diseases, which can broadly include conditions or phenotypic traits, and drugs, which broadly can include ligands or chemicals, generally small molecule chemicals. So three entities, sometimes we say the holy trinity of drug discovery, and the relationships among them, and annotations about them. So these are the kind of data and relationships which are represented by IDG and which have really made the efforts coherent across these diverse institutions and investigators. Genes and proteins not in the prioritized families were also studied, guided by understandings of complex multifactorial polygenic disease etiology, systems biology, and polypharmacology. That's a mouthful, but I think you probably get it. Reductionism is an important way to do science, but we are trying, I think all of us, to think in terms of systems biology and more complex understandings. So that very much informed IDG research. Despite the complexity of the challenge and related data, a simple categorization method was found to be highly useful and popular, the TDLs already mentioned, target development levels. And that's what this slide is about, TDLs. And the KC has already integrated some IDG resources, and in particular, featuring target development levels with that understanding, and as we encouraged. TDLs categorize targets into 4 levels from heterogeneous evidence measures, with highest level T-CLIN from activities in Drug Central, which are approved drugs with known mechanism of action. The bottom right figure is from the the right figure on the right is from the 2018 landmark paper, IDG paper, with statistics mapping TDLs to protein families and an expanded view of T-CLIN. Throughout the IDG program, and partly as a result of IDG efforts, proteins have progressed from T-dark to T-bio to T-chem to T-clin. This slide includes some IDG news, mostly new publications from 2023 and 2024 this year, although funding for IDG largely ended in 2023, not completely, but largely. New publications and other products continue to appear from no-cost extensions and other support. Efforts are underway to sustain and enhance key resources, so there is reason for optimism that IDG will continue for some time in some form. I am now going to pass it over to Anna, if you're ready, Anna.
Yes, I'm here.
I'll be happy to click through your slides when you say next slide. So
Great. Thanks, Jeremy. I appreciate it. Just like Jeremy did, I'd just like to iterate that the importance and the impact from IDG truly stems from the outcome of all of the IDG members, which range from the data resource generating centers to the knowledge management center to the cutting edge informatic tools awardees, working together with the resource dissemination and outreach center for facilitating and coordinating the outreach and exposure of these various outcomes from IDG. I would also like to note that outside of the IDG consortium, NIH offered additional druggable genome support in order to enhance research into these understudied areas. Those included multiple rounds of IDG-associated R03 grants, administrative supplements for the HEAL initiative, looking into associations between pain and IDG-eligible targets, as well as commercialization support via SBIR and STTR grants. Next slide, please. In the 9+ years that IDG existence, there have been over 500 publications from these IDG consortium grants, and these have elicited over 51,000 citations, which are still growing in magnitude and in impact factor. Furthermore, a collaboration was set up with the editors of the Nature Reviews drug discovery manuscripts for establishing the IDG Target Watch in 2019. These summary reports highlighted new findings for understudied targets, and in total, there were 22 manuscripts in this series with over 150 citations so far. There are even citations in the current year, 2024, of IDG target watches published in 2019, indicating that these early IDG work impact further work into these understudied targets. Additionally, part of this series included an annual publication of the analysis of novel drug targets for newly approved drugs from the FDA and other world agencies. These analyses are a part of the Drug Central database, which you'll hear later about, and are a part of the elevation of a target into this T-clin classification that Jeremy just explained. Next slide, please. And to expand the exposure of IDG, we also established the eIDG webinar series with presentations from IDG members and invited speakers. And over the 5 semesters of these webinar series, we had consistent attendance from all over the world. I would like to especially point out a speaker during the 2023 series, Dr. Nicoletta Landsberger, a professor from the University of Milan. She was a recipient of an Italian Research Foundation grant. The grant call was based on researchers utilizing TDL, created by KMC, for defining understudied targets, such as T-dark, and finding the connections of these T-dark targets with rare diseases, indicating the impact of KMC work outside of the United States. Next slide, please. Additional IDG outreach events were organized with other groups with the hope of enhancing collaborations and extending research into these understudied areas. So IDG held meetings with a number of different public-private partnerships, which has members from academic institutions, biotech and pharmaceutical industries. And these meetings included meetings with Open Targets, ATOM, which is the Accelerated Therapeutics for Opportunities in Medicine, and the Target 2035, which is an extension of the Structural Genomics Consortium. We also hosted invited speakers from the EUB Open and IMI, which is the Innovative Medicines Initiative. As well, there were 2 IgG-centric FACET meetings organized by IgG members. In total, these events were attended by over 2,500 people and strengthened the resolve to investigate these understudied areas. Next slide, please. Another venue, just like it is done here, is that IgG influenced and had outreach is via our YouTube channel, the Druggable Genome IgG. It was here that we posted recordings of presentations and demonstrations from various events. And to date, we have over 300 hours watched by 4,500 viewers, and the subscribers continue to increase, indicating a continual IgG interest. Next slide, please. Before I let you hear more about the data resources generated by IgG, I'd like to point out the IgG reagent resources and their impact to date. These are resources for proteins on the IgG target list only, namely that subset list of the 3 druggable protein families, the kinases, GPCRs, and ion channels. Annotations of these resources and data types were submitted into the resource management system set up by the University of Miami. And this graph depicts the magnitude of the resources based on type. To date, there were almost 1,000 records with close to 400 with complete annotations, which we deem Pharos ready and are shown in Pharos. Many resources have also been deposited with commercial vendors such as Addgene and Millipore Sigma for ease of distribution. Furthermore, IgG was an early adapter of the research resource identifiers, those RRIDs. Hence, we can now track publications utilizing these IgG resources via SiteCrunch. And to date, there are almost 300 publications mentioning IgG resources and/or projects. And we hope that continues to increase. With that, I thank you for your attention, and I pass it back to Jeremy.
Thank you, Anna. And so that was part 1, and really an overview of what has been accomplished in 10 plus years. And it is a lot. Okay, part 2 is about IDG data resources. And over its 10 years, IDG produced a diverse array of data resources and discovery tools from a correspondingly diverse team of investigators and developers, merging expertise in foundational biology and medicine with informatics and computational methodology. The graphic here illustrates a sampling of these resources. From my own lab at Mount Sinai School of Medicine, there's GeneShot, DrugShot, Apaters, and ArchES4, and the Gene and Drug Landing Page Aggregator. The Clinical Kinase Index was developed by the Stefan Schurer Lab at the University of Miami. Prokino is a protein kinase ontology developed by the Kanan and Kochut labs at University of Georgia. Reactome IDG was developed by the Reactome investigators. Here at the University of New Mexico, we developed TINX, TIGA, and Drug Central, which Jamie Holmes will describe in a moment. And Pharos, the IDG data portal, was developed at NIH, NCATS, and will be described by Tim Shields in a few minutes. Another partial listing of IDG data resources includes a few more, and with links to additional information or the resources themselves. And yes, we will be sharing these slides in cooperation with the KC. So you can click and investigate more from this slide. Pharos, as the IDG data portal, integrates many of these resources for convenient and coherent access. So I tried to highlight those with the brighter or wider borders. Pharos in many ways sits on top and integrates many of the others. We don't have time in this webinar to describe all of these by any means and more IDG resources, but it is important to understand that all are designed to serve the same overall scientific use case, illuminating the understudied genome for potential new drug targets. and in a general but very accurate sense, providing evidence from heterogeneous and independent, high-confidence sources to associate diseases or conditions with genes and proteins, which are hypothesized targets for novel pharmaceutical therapies, whether new chemical entities or repurposed drugs. So yes, there's a lot, but it's coherent and it serves a unified purpose in so many ways. Before turning it over to Jamie Holmes to describe Drug Central, I'll first briefly describe another featured and exemplar IDG data resource, TINEX, Target Importance and Novelty Explorer. One reason for including TINEX today is that there's a brand new publication just this week describing the latest version of TINEX. The first author is Vincent Metzger, postdoc at UNM and project manager for TINEX since 2021. The initial idea and prototype for TINEX was developed by Christian Beloga of UNM. TINEX briefly defines two statistics from bibliometric measures derived from the PubMed corpus via text mining, that is named entity recognition, and results from the lab of Lars Joel Jensen of the Novo Nordisk Center for Protein Research and the University of Copenhagen in Denmark. Novelty, one of those statistics, is a measure of the scarcity of mentions of a gene or disease, and importance measures the co-occurrence of a disease-gene pair, hence strengthening our belief in a Bayesian sense in that association or relationship. Via scatter plots and ranked lists, TINEX provides literature-based hypotheses with provenance to the specific publications. The new publication about TINEX includes a description of new data and features and examples of citations and usage by other investigators, indicating TINEX's use and value. Okay, I think if Jamie, if you're ready, it's time to hand it over to you for a description of Drug Central, another exemplar and featured IDG knowledge base. Are you there?
Yeah, I am. Thanks, Jeremy. I have you cannot start screen share while the other participants is sharing.
Yeah, I just stopped, so please see if you
Okay,
can.
great. Welcome, everyone. My name is Jamie Holmes. I'm the lead programmer for Drug Central. My background's in computer science, software engineering, UI and UX. I've been at the University of New Mexico for over 14 years and part of the IDG HSC Health Science Project. So Drug Central, it's a drug resource that the user can search veterinarian drugs, adverse events, indications and contraindications of drugs. They can enter a drug target, a UniProt ID, and they also can search by structure and do a similarity search on smiles. So on our download section here on Drug Central as well, you can download the SQL Postgres database. We have public and published versions of Drug Central at nucleic acid research in 2017, 19, 21, and 23. So let's go ahead and give you an overview as how Drug Central has grown over the time. So in 2017, we had drug approvals, external references, indications and contraindications. We had target annotations and target classifications. For our drug products, we were accruing drug labels, marketing status, and route. Then in 2019, we incorporated FAIR's adverse event reporting system data, including L1000 genes and target development levels, which Jeremy mentioned earlier, and FDA Orange Book data. In 2021, we added drug repurposing categories. We added sex-based separation of FAIRs and Redial 2020, an application utilizing machine learning for COVID-19. Then in 2023, we added veterinarian drugs, off-label usage as well. Can you still see the video? Can
You mean, I see your screen.
Okay. So here you go. Can you see this here, this slide?
It doesn't seem to be moving.
Okay, so this is the next slide. Sorry about that. This is a new feature that we're going to be putting into Drug Central, an AI avatar, which will be able to explain drug types, bioactivity data. It will assist with drug searches and add AI capabilities to user searches. So let me go ahead and boot this AI avatar up. This AI avatar was created with ChatGPT 4.0. And I named this avatar Ion.
Hi, my name is Ian. I'm an AI avatar created by Jamie Holmes. It's nice to meet all of you. Today, I will help guide us through this presentation. We have several important topics to cover for drugs and trawl. I will be discussing
We can't see your avatar, Jamie.
bioactivity data, data integration. We'll examine adverse drug events,
Oh, you
status,
can't?
trends, and observations.
No, we still see the Drug Central growth slide.
Interesting. Let me go ahead and share this again.
Yeah, maybe unshare and reshare.
Yeah. Were you able to see the slides?
Yeah, yeah.
Okay, let me go ahead and boot this up again. I apologize. Can you see this?
Molecular properties and drug.
Can you
I'm
see
an
it?
AI avatar created.
Yeah, we got him now.
Okay, so here we are. So this is a new feature we want to incorporate into Drug Central 2025. And I'll go ahead and boot up this avatar.
Will help guide us through this presentation. We have several important topics to cover for Drug Central. I will be discussing drug types, bioactivity data, data integration. We'll examine adverse drug events, status, trends, and observations. Let's get started. Let's take a closer look at the data curation and integration process we use to compile the Drug Central database. The process involves several steps to ensure accurate and up-to-date information. Punctual updates and data preparation, steps 1 through 3. We update tables, data sources, molecular properties, and drug repositioning categories to keep our database current. External identifiers, step 4. We integrate various identifiers such as MESH, PubChem, CID, IUPHAR, Drug Bank, UNIA, and more to ensure comprehensive data coverage. Pharmacological classes, step 5. We use sources like Daylimed RX, Daylimed OTC, and Orange Book to classify drugs pharmacologically. Drug products, step 6. Information from Daylimed RX, Daylimed OTC, and Orange Book is used to update drug product data. FAERS data, step 7. We incorporate OpenFDA data and compute LLR descriptors for male, female, and neutral populations to monitor adverse events. Bioactivity data, step 8. We update targets and activities using data from IUPHAR and Chembl to provide detailed bioactivity information. ATC code and files, step 9. Finally, we map drugs to ATC codes, provide summaries, download files, and exchange files for TCRD. This comprehensive approach ensures that our data is accurate, up-to-date, and highly useful for various applications. In this slide, I'll talk about the latest updates of the main features and functionalities. I'll start with drug types. The current Drug Central version, available online, comprises 4,995 drugs. The vast majority are small organic molecules, roughly 85% of the entries, while overall the other categories account for 15%. The same trend is followed by the orphan drugs, representing roughly 9% of the drugs in DC. Regarding the number of newly approved drugs, 2023 has been monitored up to August. For the previous years, steady trend in 2019, 2021, and 2023 can be observed, with a slightly larger number of drugs indexed in 2020. This slide presents a summary of bioactivity data in Drug Central from various sources, showcasing the number of activity points, drugs, and targets. The total activity points amount to 20,978, covering 2,754 drugs and 3,205 targets. The data sources include Chembl, HuBMAP, Drug Matrix, IUPHAR, Scientific Literature, PDSP, and drug labels, with mechanisms of action data provided in parentheses. In our Drug Central database, we have 3,205 drug targets, 733 MOA targets, 606 of which are of human origin, 1,933 drugs. The chart below shows the distribution of drug targets and drugs across various classes, including enzymes, kinases, GPCRs, ion channels, transporters, and others. Regarding target development levels, TDL, we have annotated 1,635 targets. T-Clin, 564. T-Chem, 738. T-Bio, 312. T-Dark, 21. This classification helps us understand the maturity and knowledge depth of each target, guiding research and development efforts. In this slide, I'll briefly explain the target development levels, TDL, for drug targets in Drug Central. 1T, Clin. These targets have activities in Drug Central with known mechanisms of action and are associated with approved drugs. To T-Chem, these targets have activities in Chembl or Drug Central that meet specific activity thresholds. Some targets are manually curated based on small molecule activities from various sources. 3T Bio. These targets lack known drug or small molecule activities that meet activity thresholds. However, they satisfy one or more of the following. 1. Meet criteria above the T-Dark cutoff. 2. Have gene ontology annotations with experimental evidence. 3. Have confirmed OM-IM phenotypes. 4. T-Dark. These targets are poorly understood with minimal known activities. They meet 2 or more of the following criteria. 1. A PubMed text mining score from Jensen Lab. 2.3. Gene RIFs. 3.50 antibodies available according to Antibodypedia. This classification helps in understanding the different levels of knowledge and research available for various drug targets. Now let's review some Drug Central data. Trend overview. The line graph illustrates the yearly number of first-time drug approvals from 2017 to 2021. There is a clear upward trend, indicating an increase in the number of new drug approvals. Key data points. The number of approved drugs is generally increased each year, with a noticeable peak around 2020. The dotted line represents the 10-year moving average, which also shows a consistent upward trend, confirming the growth in drug approvals over a longer period. This positive trend reflects advancements in drug development and regulatory efficiency, suggesting a robust pipeline for new therapies and treatments. This upward trend in drug approvals is a promising indicator for future innovations in healthcare. Now let's discuss Drug Central adverse drug events data. The dataset in Drug Central includes significant adverse events that have been reported, providing a valuable resource for identifying patterns and potential risks associated with different drugs. The categorization by sex and age allows for more targeted analysis and better understanding of how adverse events may differ across these groups. Indications: 1,045, with 258 unique indications. Species covered: 34. Products: 1,492. Chemical structures: 328. Synonyms: extensive list to ensure comprehensive coverage. Venn diagram insight illustrates the overlap and distinction between veterinary, 154 unique, and human drugs, 4,531 unique, with 240 to drugs common to both categories. Significance: first open access resource for veterinary medicines, enhancing accessibility and utility for veterinary professionals. We look forward to further enhancing our database to support both human and veterinary healthcare needs. In this slide, the chart highlights the significant presence of human drugs across all categories, but also showcases the essential role of veterinary drugs, particularly in the prescription (RX) segment. Understanding these distributions is vital for optimizing drug development and ensuring that both human and veterinary healthcare needs are adequately met. This slide lists the key publications related to Drug Central, highlighting significant advancements and research contributions in the field of drug discovery and repurposing. These works, published between 2019 and 2023, cover topics such as novel drug targets, off-label drug usage, and the integration of human and veterinary drugs, underscoring Drug Central's pivotal role in supporting clinical and molecular drug research. The Drug Central website also has 85,000 users annually, according to our Google Analytics GA4 data, and continues to grow. As we look ahead to 2025 for Drug Central, there are several key areas of focus. Drugs in the latest scientific papers. We will integrate the most recent finding from scientific research to keep our database up to date. Drugs in open clinical trials. We will include information on drugs currently being tested in clinical trials to provide a comprehensive view of ongoing research. Redo disease components. We plan to reevaluate and update the disease components to ensure accuracy and relevance. Update LINCS gene chemical. We'll be updating the LINCS dataset to include the latest gene and chemical interactions. Proteomic drug target interactions. We aim to enhance our database with proteomic data on drug target interactions, leveraging resources like DeepCoverMoa. Add formulations from EMA. Finally, we will incorporate drug formulations from the European Medicines Agency to expand our international coverage. And I will be added to the Drug Central UI to assist users to run advanced queries on the Drug Central database to help with drug discovery. Yes, that is right. I will help run advanced queries on the Drug Central database to help with drug discovery and use my AI capabilities to help all Drug Central users. These initiatives will ensure that Drug Central remains a leading resource for drug information. Jamie and I can answer any questions at the end of this webinar. Thank you once again, Jamie Holmes, for allowing me to assist with the Drug Central presentation and for programming my avatar, which you have named Ian. I look forward to collaborating with all of you in the future. Ian, signing off for now.
Well, thank you for that. Sorry for that technical issue. That's just a prototype of what we want to implement on the next version of Drug Central. So I'll go ahead and stop sharing my screen and hand it back over to Jeremy.
Well, thank you, Jamie Holmes, and thank you, Mr. Avatar. We'll pass the screen over to Tim Shields now of NCATS to present Pharos, the IDG data portal. All set, Tim?
Yep. Unfortunately for everybody, I'm not an avatar, so you gotta look at me while I'm talking. So my name's Tim Shields. I'm a developer at NCATS Informatics Core, and I did actually started early on. One of my first days at NCATS was at a meeting discussing whether or not we should start with the IDG program. So I was partially involved for a little bit for a while, among other projects. So I was kind of always lurking in the background as a silent partner for Trung and Raj, and then took over and then handed it off to Keith, who's also on the call, kind of heckling me on Slack in the background. So we're going to talk about Pharos, which we called the open source target illumination platform, and it was designed to be the front end, the user interface for the TCRD, which in itself was the entire bolus of data that the IDG had aggregated and brought together to try to develop these target development levels. So the KMC, the Knowledge Management Center, provided, besides Drug Central, the Target Central Resource Database and Pharos. So the database itself aggregated 78 resources, has expression data, publication data. There's a whole list. I don't need to read all of them. And then with this data, use different measurements that were available to determine the development level, which Jeremy and Anna have already spoken about, based on dark targets have little known information to TCLIN targets where there is an approved drug available. From there, NCATS Informatics Group designed a user interface, because having all this data is nice, but people did want to be able to browse it and view it and kind of see what's inside this database, and then potentially gain further access. So a little over 20,000 targets, a little under 20,000 diseases, whole bunch of ligands and compounds. From all of this data and the different sources, we created a large list of different filters that were available, added some user functionality to be able to sign in and upload and save lists, share that information, and then as the program progressed, We performed user research, and we're always giving demos and seminars to try to figure out what other aspects people were interested in seeing and enhance that, in addition to working with data generating centers within the IDG that were also generating new information. So we tried to incorporate as much stuff as possible. Here's just a list of some of the different data sets that were in the TCRD and are currently available with the package. So with all of this information, we wanted to operate Pharos as almost a shopping site where you're going to have information you want to find. You're going to want to get a list of targets related to that information, and from that, see what is available to them, maybe compare development levels, see if there's any commonalities, and hopefully potentially find targets that aren't necessarily obviously associated with your target, but may share commonalities and could be a target of interest. So we have the ability to search over a multitude of facets and different properties. And I'm going to move through these pretty quick. You know, obviously you can browse. Browsing through the entire genome in Pharos is not too helpful, but there's filters along the side that allow users to kind of stack different categories and different filters and find hopefully targets that they're interested in. From the filters, we have different types of filters. You can search within the filters themselves. Each one has definitions. We made a pretty strong effort to include help and definition panels wherever possible. You can filter by numerical properties, so novelty or a PubMed score, how much published data there is about it. And so really the idea is to take these 20,000 genes and using similar properties, narrow it down to hopefully a workable set. Each drug or target details page is divided into sections with a general overview, if there's approved drugs or ligands or active ligands available, some of the different disease associations, disease novelty. This is the 10X that Jeremy had brought up. There's pathway information. We worked with Reactome to incorporate their data as well. There's wiki pathway information. We pulled in AlphaFold models and used some PDB models to display information as well, the structures. You can filter and search and examine gene ontology terms. And so, you know, each page just has tons of information that's available for users. Protein-protein interaction, we have the sequence details. Yeah, it's a pretty long list going through each page. So to help with that information, there's going to be this question mark button that will explain where definitions come from, what the source is, and how to get more information. So we talked about the target development level. So this is just kind of a quick example of how a target progresses from T-dark, what the requirements are, what the cutoffs are, how to view the supporting data, and then how to get all the way to the targets with approved drugs. And then, yeah, this is a panel that pops out. It'll show you the definition. If there's more information about how a score was computed, we'll have that as well. So again, trying to really highlight where this information, where these measurements are coming from. And I mentioned IDG generated resources. So these are all groups that were part of the IDG. And this data is unique, you know, right now to the IDG. So it's information that you can find here and get, you know, reagents, assays, cell lines, mouse models, different data sets that were all generated as part of the IDG. In addition to your list of targets that may be associated with the disease or have a drug approved for them, you can also find targets that interact with them. And as you perform some of this list analysis and compare different lists of targets, we have dynamic sections that'll show up to show, you know, what the interaction details may be, what the association may be, what the activity may be, and it's you know, varies depending on what properties of the list that you're looking for. You can find targets that are similar based on different processes. So right now, this one's just the Go process, but there's a bunch of different categories that you can then try to find targets that have similar values. And then you can, again, you can read the common values, you can read the data that gives us these results. All of these lists are downloadable, so you can save a lot of this target information as well. And that's, yeah, we also updated some of the publication and components. So we've got our word cloud, an improved publication view. You can download publications associated with the target or the gene riffs associated with them. And then it'll also show you how recently this information was downloaded. We added new visualizations for the 10x data. We just had in PeerJ the 10x version 3 publication come out. And so this allows you to kind of highlight where your target is or where a group of targets related to this disease novelty is. So it's a really helpful way to take something that's a scatter plot and see kind of, it gives you a little bit more information about what you're seeing within that plot and a little bit more details about what it is that you're looking at. But you know, the whole purpose from this though is we have this massive database and we'll show people all this stuff and they'll say, well, you know, that looks great, but where can I get the data? So people are you know, and especially even at NCATS for informatics, people just want the data. So we have lots of different ways you're able to get the data out as well. If you just want some data about a particular target or a particular section of it, there's a download button. It brings up a download query that you can build. You can pick and choose what information, what properties you want to get. It'll show you what the SQL query that we're going to run is, and then a quick sample of what your information is going to be. So once that's done, you can run your download query and download the information. We do tell people though, if you have tons of information, the database itself is downloadable. So you can download and load it yourself. And then these SQL queries come in handy because then it gives you a starting point as well. In addition to just the raw database, we incorporated, we moved from a traditional REST API where you would get different levels of information as you progress. So to get programmatically UniProt a session for a target, if you have a UniProt ID, how can you get drug information? So first you would have to get the target data to get our ID for it. Then, and this is an old version, none of these links work anymore. But then you would have to get just the targets and then get information about each target. So it was multiple stages and it kind of started to take a while. So we switched over to GraphQL. It was created by Facebook and operates more on some of the principles of interconnected information like a graph database. So instead of having to make all these specific jumps in one query, you can have it follow this loop. So you have, you pass it an object with the query. Actually, this is a better way to look at it. With what the query is, so you can pass in parameters and you say, I want the UniProt ID, the ligand counts, the name and value, and then give me the drugs associated with that, filter out the ones that are approved drugs, and then give me the names and activities. So this would have been, you know, 1, 2, 3 queries to get that information, whereas here you can get it all in one query and page different sections of the result. So rather than us, the developers, kind of determining what information should be shared, this allows users to kind of pick and choose whatever information they want and really help streamline the process. And again, we have a detailed documentation page and a sandbox. You can try these queries out at pharos.nih.gov/API. That's all I've got. You know, as usual, there's a ton of people to thank who are involved within NCATS and with the IDG, and we've got the team itself. This was back in the fall 2023 where we had a NCATS informatics retreat. So Keith, who does you know, the majority of the development work on Pharos now, Jessica Binder, who we snagged from UNM and is now helping us work on kind of a next stage of target information, and Evie Mate, our informatics head. So I'll pass it back to the group and can answer any questions as well.
Thank you, Tim and Keith Kelleher, for just the amazing work on Pharos. over the years. It's really an impressive thing. I have one wrap-up slide, which I think we can fit in under the time limit if Mackenzie doesn't blow the whistle or something.
No, absolutely.
Okay, so basically, I think you've gotten the message, audience, for over a decade, the overall message, for over a decade, but with some work continuing, a diverse and talented consortium of experimental and computational researchers has focused their efforts on illuminating understudied human proteins as potential new targets for pharmaceutical therapeutics, using a wide variety of methods and approaches. The resources generated are available to continue facilitating and accelerating discovery, with efforts underway to sustain these resources to continue and maximize their benefits. For those of you wishing to learn more about IDG, these links will lead to an extensive set of documentation, publications, and the resources themselves. Please also feel free to contact me, and I believe the others on this panel and other IDG contributors to learn more and perhaps explore making effective use of IDG data resources for your research. And to echo the others, thanks to all the IDG leaders, contributors, and advisors over the years whose enlightened and dedicated efforts produce these resources, pun intended. So thanks, everyone.
Thank you all so much. Before we leave, again, thank you so much for IDG for participating today. I just wanted to highlight our next webinar will feature an overview of the Human Reference Atlas version 2.1 from HuBMAP. So please tune in. And if you are unable to attend either part of this or you have colleagues that haven't been able to attend today, we'll have this recording up on YouTube along with the coordinating slides. So thank you again for everyone at IDG for participating. Thanks, everyone.
Yeah.